All right, well, I think I'm going to start, and I started the recording so other people can uh, follow later on. So the topic of the month is methylation, and hopefully you can see that screen there. So, and this is me here. And why don't we just get started and talk about methylation? So, and let's see if we can't get this going here. All right, so let's start out. What is methylation? So methylation is adding a methyl group. And because we're all carbon life forms, then it's important to be able to move methyl groups or carbons from one molecule to another fairly easily. And there's quite a few of these reactions going on inside the body. And so it's basically moving a methyl group around. And what is methyl? Like I said, it's a carbon group. So C carbon usually has 4 hydrogens that can attach. But when you're going to move it around, you need one spot open to attach it to. So methyl group is CH3. CH4 is called methane. And the term methyl comes from the Greek. It means wood wine or wine wood. And they found that just regular wood would turn into a wine. We call that wine now, we call it methanol or methyl alcohol. And there's other kinds of alcohols as well. You may have heard of methylated spirits or denatured spirits. Those are a little differently. Those are going to be regular alcohol products that you find around the house that you don't want kids drinking, like maybe Listerine. Well, no, not Listerine. Maybe something like some kind of cleaners or things like that that have alcohol in them. And this is your reaction. Basically, you have some item, some molecule R that you want to methylate. So there's a donor, and the donor transfers the methyl to the R. And there's an enzyme that makes this happen. That's called methyltransferase. So you could think of it as the R is getting methylated, the X is getting demethylated, and there's a methyltransferase that's helping the process along. And yep, and we call those MTs, methyltransferase MTs. Okay, so give some examples of what a big difference it can make. So here's a little molecule that a lot of kids take it to help them concentrate in school. And then you see we have methylated, I just added a carbon at the end there. And these are two different molecules. The one on the left is amphetamine, which is available by prescription and is fairly safe. And the other one here is meth, which is illegal. So these methyl groups can make a big difference. So now the question is, where do we get our methyl from? So for people that, normal people, let's say, that aren't in the hospital, they're going to get it from their diet. And we get about 50 micromolars of methyl each day. And where does that come from? So for the most part, it's going to come from choline, methionine, and folate. Those are really the 3 main areas that we'll get our methyl from. Methionine, you may know, is one of the amino acids, so that's going to come from protein. And folate is kind of a B vitamin, and everything has folate now. The past 20 years, it's fortified in bread and lots of other things. So folate is fairly easy to get. It's also in foliage like greens and so on. So Talking about folate, let's go into one of the main carriers that we're going to be discussing today, tetrahydrofolate. So tetrahydrofolate just means that the folate has 4 hydrogens attached to it. And so folate itself, like I said, it's kind of a vitamin. We call it vitamin B9, and it's available in food. And I don't have a picture of it quite yet, but it has a pterine group, which is like a double ring, and then it has a glutamate at the end. And so folate gets converted by this enzyme dihydrofolate reductase to folate dihydro and tetrahydro. So the tetrahydro is the one that's more active and gets to be added onto it. And where does it get added onto? It's this pterine ring that I mentioned. So it's got room to be methylated twice, one at the nitrogen 5 position and one at the nitrogen 10 position. And so if you add one methyl group on, it's methyl tetrahydrofolate. If you add 2 of them, it's going to be methylene tetrahydrofolate. So here's a little picture of everything that's going on. So we'll come back to this later. But if you start up in the upper left-hand corner, we see that folate is here. That's dietary source. And then this enzyme I talked about, dihydrofolate reductase, acts on it twice to get you to dihydrofolate and then tetrahydrofolate. And anywhere you see NAD, that's niacin. So that's going to be vitamin B3. And any place you see FAD, that's a flavonoid. So that's like riboflavin. That's vitamin B2. So all the vitamin Bs are kind of showing up in here. And for this step right now, we're just kind of talking about what happens to folate. It ends up as tetrahydrofolate. And I did mention that you could convert that into, what are you seeing? All right, let me take a look and see if what is being shared. Yep. So, so can you see it now? I think we're up to slide 10. So, thank you. So anyway, once you get to tetrahydrofolate, like I said, you can add carbons to it. It can be methylated twice, the N5 and N10. And this product here, which is the methylated folate, is really the center of what's going on here. And there's 4 things that this can happen, what can happen to, and we'll discuss those later. All right, so here's another picture, just a little simplified to make it easier to see. And on this picture, I want you to focus on the boldface items. So we got folic acid here, or folate, which we already discussed. Then methionine, which is the amino acid that we get from our protein. That's a key player. And then I said the third thing that we get our methyl groups from is choline, and that's over there. So we got folate, methionine, and choline, the 3 big players. And then let's also in boldface is homocysteine, and that's was in the past thought to be a bad thing. Homocysteine, bad, but it's really just a sign that bad things are happening. So if you have a lot of homocysteine, then it's a risk factor and probably means that you're not getting your B vitamins. It's kind of like a symptom. So let's go back to methylation, and the 3 things we're going to focus on is what methylation can do. So it's involved in DNA. That's what I would consider the number 1 thing. Then number 2 is it's involved in the body's making things. So the body cannot make, say, certain things. It needs like ingredients, and then from those things it can make other things. And so in order to make those other things, like amino acids and neurotransmitters and adrenaline, you need to have methylation occurring. And finally, detoxification. So the liver does a lot of things to protect us. And one of the things the liver does is it methylates various things or toxins. So DNA, let's talk a little bit about DNA. So methylation is needed for cell division. In particular, we generally need the folate is a key aspect. And when this process is blocked, the methylation that causes the DNA synthesis, I think I spelled that right, then cells cannot divide. And that's a problem for an individual cell. It can't divide, but in general, it's useful for us if we have a cancer and you want to stop the cancer from growing, or if there's an ectopic pregnancy, you want to stop it from growing, or if you have some bacteria maybe, and some bad bacteria, you want to stop them from growing. So it has some good and bad things with it, blocking it. That's like the medical viewpoint is how can you use this? And this is the chemistry reaction is tetrahydrofolate gets methylated twice to form N5 and 10 methyl tetrahydrofolate. And this here basically converts UMP to TMP. So if we go back to that picture, we see that we have UMP, which is I think it's uracil monophosphate, into thymidine monophosphate. And that T there, or DTMT, that's part of DNA. And so this lower left-hand corner is the DNA synthesis part that methylation is part of. And so, so basically, you may know that we have base pairs in our DNA, and it's going to be C, G, A, and T, and thymidine is one of those. And of course, we make the other ones as well very similarly. All right, so other things that can happen to DNA is there's something called DNA modification. And so the base pairs that I talked about can get modified, and that happens usually after birth or after the cells divide, if it's bacteria or cancer. And this means epigenetic modification, means after the genes are copied, different things happen to those base pairs. So you can get acetylated, and you can get methylated. And when it gets methylated, in fact, there's many, many large portions called CGP islands or GCP islands, where you have many, many repeats, all of them being methylated. And so these methylations can cause the genes to be turned on and off, and each individual cell, if that happens, can progress in different directions. And these changes are now thought to be causing, or too much of these, cause autism, cancer, anxiety, heart disease, and perhaps a couple of others. So too much methylation could be a bad thing. That's current thinking. Okay, I said I was going to show you some pictures of these. This is methyl tetrahydrofolate. So this is folate, basically, vitamin B9. You see it's got a double ring over here. And then there's a place for the methyl group to come right in there. So that's the methyl carrier tetrahydrofolate. And then if we go back down to the bottom left, this is methionine here. Methionine has the sulfur. It's one of two of the amino acids that has sulfur in it. And right attached to sulfur is this methyl group. And what you do is when you attach adenosine to it, it makes, attached to the sulfur, it makes this bond from the sulfur to the methyl group unstable, and this carbon can now fall off very easily. So this methionine, or the SAM, is also a donor. And then I said choline, and then B12 also is involved, but B12 is a little more complicated. So just a quick rundown of some of the vitamins that are involved. Okay, so we already talked about B2 and B3. Those are everywhere in almost every reaction. And B4 is choline. So we talked about choline is the main methyl donor in methyl that we get from food. Then B6, pyridoxal phosphate, that's important for some reactions. B9, which is folate, is another important food source for methyl groups. And B12, or cyanocobalamin, that one is also involved. So those are a lot of vitamin B's involved. And then the amino acids, methionine, I already talked about. Cysteine is very similar to homocysteine, so it's kind of a precursor of methionine. And then serine and glycine are involved as well. Okay, so what else is methylation involved in? So aside from DNA, it's involved in making things inside the body. So everywhere you see an MT here, that's an enzyme in the body that transfers methyl group from one place to another. And so there's one very big methyl transferase that's important, and that's this one here. It turns serine into glycine. And it's not important per se that we want to change serine into glycine, but that's kind of a requirement for part of the other stuff that's going on. And this enzyme requires vitamin B6 and iron. So if we go back to the picture, I'll show you where this reaction is important. Let's go back here. It's right here. So remember I said tetrahydrofolate gets methylated to this key player in the middle. Well, in order for that to happen, the same enzyme basically has to convert serine to glycine. And I said already that that involves vitamin B6 and iron. And so that's why this is an important reaction. This methyltransferase is involved in that methylation here, but it has to, how to say it, has to get the methyl group from somewhere. And so that's our first methyltransferase reaction. I'm not going to go into detail about a lot of these, but just for your knowledge, serotonin gets converted to melatonin. That's a methyltransferase. We have methyltransferase that will work. They'll act on nicotinamide, that's vitamin B3, tryptamine, that's involved in serotonin as well, I think. Histamine is involved with things that itch. And norepinephrine to epinephrine, that's how adrenaline is made, one way adrenaline is made. And then we have ASMT, it's AS3MT, not to confuse with the ASMT. This enzyme AS3MT not really belong in this slide. It's just to show you that when we get arsenic in our body, this actually happens in the liver. Most likely, we can detoxify it by adding a methyl group to your arsenic, so it's less toxic to us. And I'm not sure if you can see the bottom row, but COMT will convert dopamine to homovanillic acid. And so this one is an important one, so I wanted to say something about COMT. COMT stands for catechol O. So every methyltransferase can either be an N-methyltransferase attached to a nitrogen. We saw where it can attach to a sulfur. It can attach to an oxygen as well. So here we go. COMT uses magnesium. And like I said, it converts dopamine into something. So the first thing it converts it to is into 3-methyl tyrosine, I guess. And so So that's an important thing, especially in the brain, to have something that gets rid of dopamine so it doesn't stick around too much. But on the other hand, you don't want to get rid of all of it because dopamine's important. And it's found that this enzyme, COMT, has different variants. So depending on what part of the world you're born in, depending who your parents are, you can be a fast metabolizer, meaning you have both parents gave you a V variant, or you could be a normal metabolizer, or you could be half fast. And it's the fast metabolizers that have a little bit of a problem because they get rid of their dopamine at a very fast pace. And they also will turn epinephrine into metanephrine. And so they have different concentrations of these important neurotransmitters than other people. And so one of the things they find that these are the people that are increased risk after they smoke a joint of marijuana of having a bad reaction or a psychosis. And there's some other things that these people also have. I'm not sure if I'm going to talk about it, but perhaps. Here's a list of variants of this, and these RS genetic variants, the RS ones. So for example, if someone has done 23andMe, they can look up some of these and see whether they have low COMT, high COMT, or normal. And for the ones that have high COMT, like I said, could be a problem. Here's, let me just see what I said. Yeah, so that's high COMT. That gets rid of the dopamine. Now, there's also a problem with low COMT. That's what this next slide says. It says that if you have low COMT and low magnesium, then you could have a problem there too. So normal magnesium and normal COMT will create this product called I think it's 2-methyl estradiol. It has to do with estrogen, and that causes normal, safe blood pressure. Whereas if people that have this decreased COMT variant, when they have not enough magnesium, then they end up with this problem in their kidneys that gives them high blood pressure. And so this is another variation of why methyl transferases and methylation is important. Okay, so that's enough for COMT. Another, oops, let me move this over here. Another enzyme that's pretty well known is methylene tetrahydrofolate reductase. And so we already talked about methylene tetrahydrofolate. So if we go back to the picture here, remember what happened is we took tetrahydrofolate and we turned it into, we methylated it twice. Now there's an enzyme called methylene tetrahydrofolate reductase that takes this tetrahydrofolate that's methylated twice and gets rid of one of those methylations, along with vitamin B2 and B3. And this gene has variants also. And so let's see what happens if this gene works a lot. Then it produces more of this 5-methyl, just regular methyl tetrahydrofolate. And then if you look at the right-hand side of this diagram, which we haven't really looked at yet, this here will then push this reaction. It'll go in this direction where this arrow is, and it'll turn the methyl tetrahydrofolate back to tetrahydrofolate. So we have that again. But while it's doing that, it turns homocysteine into methionine. And so, and that uses vitamin B12. So there's 2 reactions in the body where vitamin B12 is used, and this is one of them. And so I said homocysteine is kind of a bad thing to have too much of. And so some people used to think that having this gene working is a good thing because it gets rid of the homocysteine and it gets you back to your methionine methyl donor. So conversely, if this gene is not working very well, then you have less of this process going on and you can have some homocysteine buildup. So let's take a look at that methylene tetrahydrofolate reductase gene, several pages ahead. And I think it's over here. Yep. Okay, so this is a gene needed to process folate. Well, it's a little bit vague, process folate, but to get it back to tetrahydrofolate, to get it back to the beginning of that cycle. But while it also does that, it will convert your homocysteine back to methionine. So it gets rid of the homocysteine. So in this gene, again, depending on where you're born, your parents and so on the 677th base pair, you could either have 2 Cs, or you could have 1 C, or you could have no Cs. And so normal would be having 2 Cs. And the variant, which would be a little bit bad, would be to have 2 Ts. And about 11% of the humans have this TT variant. And so what does it mean that they have this TT variant? Well, it means that both of their genes are somewhat defective. And what does defective means? It means that they have their protein has changed one of their amino acids from alanine to valine, and that makes it less active. So I forget how much less active, maybe 30% or something, but being less active in both genes means that you're going to have the imbalance in that cycle that I showed you. And so what will happen is you're going to accumulate homocysteine. Maybe not a lot at first, but if you have some stressors or other things going on, you could accumulate a bit of homocysteine. And so homocysteine, like I said, is a risk factor for heart disease and stroke. That was the, at least, way of thinking for a long time. So the risk factor is for heart disease and stroke. It's been shown that if you have homocysteine, you're likely also to have increased blood pressure, cholesterol, high sensitivity CRP, and sugar for diabetes. And so this gene, you can find out once and for all what you are, CC, CT, or TT. And so CC and CT is not a big deal, but TT has a little bit of a risk. There's another variation called in 1298 that also 11% of people have. That's not as serious, but just so you know, there's many variants, but the variants that affect a large number of population, 11% of the population to be exact, those are the 2 that are involved that are worth knowing about. All right, so I don't know how much time I'm going to have because I want to leave something for questions. So let me just briefly see what else we can talk about. The third topic I said was your detoxification. So your liver is going to detoxify things. The first pass is not really relevant to methylation. It's just kind of like an antioxidant. It's basically going to oxidize. Let me just see. It's the second pass that's more important, and that involves methylation. And so a lot of things that go through your liver on a second pass will get changed, altered, and methylation is one of the things that can happen. So, for example, I think I pointed out arsenic gets methylated in the liver, and then there's going to be certain medications as well. And then let me see what else. Choline, yeah. So choline, I mentioned, is not only an important source of methyl groups, but rats that are fed that are deficient in choline and methionine, not only do they develop fatty liver, but their liver gets completely shot, fibrosis and liver cancer. And so, so it's important to get your methyl groups. But on the other hand, you don't want to get too much. And I think I'll stop there for any questions. Let me get back to the picture of what's going on, just so we can have that. And why don't we start off? Anybody have any questions? Let's see. I guess you missed, the slides weren't being shown at the very beginning. So why don't I just show you at the beginning what I was talking about. So this here is the pill that the kids take for school kids to help them concentrate. And this here I showed was just a methylated picture. So if you didn't see that picture, I can show you that now. And so this here is amphetamine, and this is methylamphetamine or methamphetamine. And meth is a bad drug that causes lots of problems, especially in the brain and the teeth. And amphetamine will help for ADHD. Okay. Let me see what, I didn't talk about SAM too much. SAM is basically methionine that when you attach the adenosine, it makes the methyl group easier to fall off. And then SAM turns into S-adenyl homocysteine. Methionine does produce homocysteine in this cycle, and you need the vitamin B12 and this methylene tetrahydrofolate reductase to get rid of the homocysteine over there. And some people will talk about a SAM to SAH ratio. But I don't pay too much attention to that. What kind of questions might people have? What kind of vitamins do I recommend? I don't really, not really big on recommending things, but for vitamins, I take vitamin D myself. And then I do believe the choline inositol pathway is important. So inositol is a very cheap, easy thing to get, and it tastes like sugar, and it doesn't have any calories, and it's very good for a lot of things in the body, but I can't really recommend it just because I take it. So we got about 4 minutes left. Nobody have any questions? You can send it in the chat anonymously, and I can answer anything from the chat. Guess we could talk a little bit about this MTHFR gene. And so, for example, let's say you got tested for this and you found out which one you were when you were a kid. Would you need to be tested again when you're 50 years old or 60 years old? Well, no, unless you had like a bone marrow transplant or something, your genes is going to be the same. And if you were born as a TT or a CC, that's going to be the same forever. And this is the way you would find out for this is you would do a molecular test. okay? And molecular test means generally you're going to do some PCR. You're going to take a little piece of the genome from the person, usually from their white blood cells, but could be from a swab or saliva. And then you're going to break that down, mix it with primers for the known T variant. And then you're going to amplify over cycles and cycles and cycles. And then you're going to attach a little green dye to what you've tagged to see if there's anything there. Then you wash out everything that's not tagged. And now you measure to see how much of your glow in the dark green, let's say, is there. And that will tell you if you have your T. If you have a little bit of T, means it's mixed. If you have a lot of T, then you have the variant. And that's how we test for that. All right, so I think if nobody has any questions on this, we'll stop and feel free to ask questions later on. And I'll stick around for a couple minutes as well. So I will see you later. Goodbye. Oreo cookies. How about orcas? That's a joke for crossword puzzle people. They ask, what is black and white? And starts OR blank blank. And so if someone says it's Oreo is black and white, starts OR blank blank, you can say, no, it's orca. If they say it's orca, then you say, no, it's Oreo. So that's right. The big mammals in the sea that eat great white sharks for breakfast.